Okay, so let's finish up this here. The point of this slide here is to show that at the ganglion in the autonomic nervous system, when we are synapsing from presynaptic to postsynaptic neuron, we always use acetylcholine, no matter what, regardless of whether we're on the sympathetic side or on the parasympathetic side. Now, when it comes to the synapse to the target tissue, on the sympathetic side, we will be using norepinephrine. So to go from the postganglionic neuron down to the target tissue, which of course would be cardiac muscle, smooth muscle, or a gland, we're always using norepinephrine on the sympathetic side. On the parasympathetic side, we will be using acetylcholine again to synapse from the postganglionic neuron to the target tissue, which again would be smooth muscle, cardiac muscle, or a gland. One last thing to note would be that the acetylcholine receptors at the ganglion are nicotinic type of acetylcholine receptors. That means they're ion channel type of acetylcholine receptors, versus the acetylcholine receptors here from the postganglionic neuron to the target tissue. They are muscarinic type of acetylcholine receptors. Now you might wonder how this would work if we're dropping a neurotransmitter onto, let's say, smooth muscle. And you should remember from anatomy that smooth muscle tissue, those muscle cells, smooth muscle cells, they're relatively small. So how do we ensure that smooth muscle tissue then sort of as a whole organ will contract? Think about it. When a baby needs to be born, it wouldn't help if the uterus has a little twitch here and there. You need to have these peristaltic wave-like strong contractions to push the baby out. Or same is true for the GI tract. Well, how do you achieve these smooth area kind of contractions that will move stuff along? The answer to that is that we have these varicosities. That means the autonomic axon will run into the tissue, and it has these little bulges. They're here enlarged. You can see these sort of bubbles, almost these bulges, and they contain neurotransmitter. So that if you are running this axon here through the tissue and have these little varicosities that contain neurotransmitter, then you could actually simultaneously release the neurotransmitter, dump it on the entire area right here, and achieve a whole area contraction of smooth muscle cells. So let's take a closer look even how that works. So the action potential would be coming down here, this autonomic axon, and then here we are arriving at the target tissue. So again, the target tissue would be smooth muscle, cardiac muscle, or gland. And anyway, so let's pretend we're smooth muscle. So the action potential is coming down. We're opening voltage-gated calcium channels, just like we had at the other synapses that we talked about in the nervous system lecture. And that causes, in this case here, we are apparently on the sympathetic side, so we're releasing norepinephrine from these vesicles, so the neurotransmitter will be released. Then the norepinephrine in this case will travel across the synaptic cleft, will bind to the adrenergic receptor, and then we get the response from the target cell. After the norepinephrine has done its job, it needs to either diffuse away or be actively transported back for recycling purposes. Now here briefly one more time in steps, the whole procedure. The action potential would be arriving here at one of these varicosities that would open voltage-gated calcium channels, which then calcium diffuses into this bulge part of this autonomic axon. And now we are releasing norepinephrine. That's our neurotransmitter. The norepinephrine will be released right here, will bind to its receptor. That's an adrenergic type of receptor. And then we get a response from the target cell. Now the job would be to remove the norepinephrine, and that can be done in a couple of different ways. It could be just that it's diffusing away, or it could be active transport, putting it back into this varicosity for recycling. There's maybe one thing that you should know. You may have heard of an enzyme that's called monoamino oxidase. That's an enzyme that will metabolize the norepinephrine and then help with its recycling. But there is a class of drugs out there that's called monoamino oxidase inhibitors, MAO inhibitors. That's an important class of drug. If you wanted to keep, in this case, the norepinephrine longer around, then these MAO inhibitors would do that job. Here you have a summary table that's showing you the neurotransmitters that are being used on the sympathetic division. We have norepinephrine as the neurotransmitter from the postganglionic neuron to the target tissue, and the receptor types would be alpha and beta receptors, alpha and beta adrenergic receptors. And on the parasympathetic division, we're always using acetylcholine, and at the synapse from the postganglionic neuron to the target tissue, we could use either nicotinic or muscarinic type of acetylcholine receptors. And the remainder of the other part of the table is not that important for you guys. Now this table here summarizes some of the subtypes of the adrenergic receptors. So adrenergic means it responds to adrenaline, which is the name that was formerly used for norepinephrine and epinephrine. So adrenergic adrenaline now refers to norepinephrine. The receptors that the norepinephrine binds to, they're still called adrenergic receptors. And we have alpha and beta receptors and with their subtypes. For alpha receptors, the main two subtypes are alpha 1 and alpha 2 receptors. And for the beta receptors, beta adrenergic receptors, we have beta 1, beta 2, and beta 3. And there are many more subtypes actually, but for the purpose of this course, you don't need to know any more than that. Okay, so on the parasympathetic side, we're always using acetylcholine, both at the ganglion and then at the synapse from the postganglionic neuron to the target tissue. At that juncture, we're using the muscarinic type of receptors, and those are all G protein coupled receptors using second messenger pathways, and there are at least 5 subtypes. And lucky for you, right now you don't have to worry about that, but if you're taking pharmacology at some point, you will have to worry about that. The adrenal medulla. We refer to the adrenal medulla as a neuroendocrine tissue, and so that's it's basically a modified ganglion. But anyway, it's the adrenal medulla. The name indicates it. It's adrenergic, so it's associated with the sympathetic branch, and the primary neurohormone that it's using is epinephrine. That's secreted into the blood, and that's what this looks like. So the adrenal medulla, so the center, medulla is always the center here. It's a modified sympathetic ganglion, and it releases, so here we have the preganglionic neuron, and so here this is sort of this modified ganglion, this gland / ganglion, and it's releasing epinephrine directly into the blood. Now here the adrenal cortex portion, the outer side here, that's a true endocrine gland, and you get hormones such as the mineral corticoid aldosterone there, or the glucocorticoid cortisol, that we'll be covering in the endocrine lecture. But here the center, the medulla region of the adrenal glands, that is a modified sympathetic ganglion. It releases epinephrine into the blood. Here you have it one more time. So we're leaving the spinal cord. We have the preganglionic sympathetic neuron going to the ganglion, and we're using acetylcholine as usual here at the ganglion, but here then we're releasing epinephrine directly into the blood. So we consider the adrenal medulla a modified sympathetic ganglion. Now this table here summarizes agonists and antagonists of neurotransmitter receptors. Well, on the cholinergic side, so we have the acetylcholine receptors. They can either be muscarinic or nicotinic, and fittingly the agonist for muscarinic receptor is muscarine, and the agonist for cholinergic acetylcholine receptor would be nicotine. Maybe as especially from a pharmacological perspective. The antagonists are often very interesting. The antagonist for muscarinic type of acetylcholine receptors will be atropine, very strong antagonist. Also strong alpha-bungarotoxin. Those are probably, and curare, we mentioned that one in the nervous system lecture. On the adrenergic side, we have the alpha and beta receptors with their subtypes. What's important here would be taking a look at the antagonists. That would be alpha blockers, and then very important class of drugs are beta blockers. They block the beta adrenergic receptor in the heart, and that's a widely used class of drugs, beta blockers. So they block beta adrenergic receptors in the heart specifically. And here, one more table. This is sort of your summary table comparing sympathetic and parasympathetic side of the autonomic nervous system. You can just study from here if you like tables. Other than that, it's just the same information, just presented in a table format, all the stuff that we was just talking about. And that concludes actually our discussion of the autonomic nervous system. And now we would be moving on to the somatic side. So that means now you are in control again. And on the efferent division, the somatic pathway, that will be those pathways that go to skeletal muscle, that of course is under your control, and that would be somatic motor pathways. There we have a single neuron, a motor neuron that leaves the central nervous system, the spinal cord, and goes all the way down to the target tissue, which is now skeletal muscle. And there we have what's called the neuromuscular junction, which will be the focus of our muscle lecture. We're releasing acetylcholine at the neuromuscular junction, and then at the muscle fibers, skeletal muscle fibers, we always have these nicotinic type of acetylcholine receptors, and we'll talk much more about those when we get the actual muscle lecture. So here, still a comparison. The autonomic pathways, so the efferent division, we have the autonomic pathways that will be including the parasympathetic side, the sympathetic side of the autonomic nervous system, and then the adrenal pathway, where we have the adrenal medulla as our modified ganglion, like we talked about. And here the different types of neurotransmitters and their receptors are listed. And then on the somatic side, the target tissue is now skeletal muscle, as opposed to the autonomic effectors are smooth muscle, cardiac muscle, and the glands. And again, you can study this table. That I don't need to say much more about this. Now, a brief intro into the somatic motor division. Again, you have a single neuron that starts in the central nervous system. It's myelinated for fast conduction, and then it ends in the neuromuscular junction. That's your synapse to the effector organ. In this case, it's skeletal muscle. We will have a very similar image to this one once we do the muscle lecture, and so I'm just not going to say much about it here. You won't hear much more about the neuromuscular junction. That will be this part here, and it's enlarged in this image right here. We're always using acetylcholine at the neuromuscular junction, and you already did a lab on that, so you kind of know some of this information. Here will be the neuromuscular junction, so the motor neuron. It starts branching out at the end, and then it will activate a number of muscle fibers, and so that would be the motor units. And well, here, take a look. This is always myelinated for fast conduction. This branching is very typical of the motor end plates right there. Here up close, the motor end plate. Again, we get much more on this when we do the skeletal muscle lecture. And here again, don't forget the neurotransmitter that's released here at the neuromuscular junction is always acetylcholine. The receptors are nicotinic type of acetylcholine receptors. And here more up close, the neuromuscular junction and the acetylcholine here in the synaptic vesicles. And then we have the these are nicotinic type of acetylcholine receptors. So here, once the actual potential arrives, we get the calcium influx that causes these vesicles to fuse and release the neurotransmitter acetylcholine, which then will bind to these nicotinic type of acetylcholine receptors, which are ion channels themselves. So nicotinic receptors, they are ion channels themselves, and then they will carry the actual potential along the muscle fiber. And again, we will see much more on that, much more detail in the muscle lecture. Here's your last slide for this lecture. The acetylcholine, actually 2 acetylcholines would have to bind to this nicotinic type of acetylcholine receptor, and it's a cation channel, and once it's opening up, it will change the membrane potential of the muscle cell, and that's ultimately going to lead to a muscle contraction. More to come in the muscle lecture.